Hi, I'm Sohail. Today I'm going to talk about cilia and flagella. In this lecture, I'm going to focus on structure and functions of cilia and flagella, difference between prokaryotic and eukaryotic flagella, motile cilia and his cousin primary cilia, and process of formation of cilia. Cytoskeleton like microtubules also make up two types of cellular appendages important for motion, that is cilia and flagella. So, cilia and flagella, they are made up of microtubules. As one science writer noted that a single cilium is a cell's eyes and nose, GPS receiver and even weather vane. Cilia is plural, cilium is singular, flagella is plural, flagellum is singular. They are hair-like outgrowths of cell membrane. Flagella are comparatively longer and responsible for cell movement. Cilia are small structures which work like oars, causing the movement of either the cell or the surrounding fluid. Length of flagella is about 200 micrometer and length of cilia is about 2 to 10 micrometer. Flagella are long hair-like structures that extend from plasma membrane and are used to move an entire cell, for example, sperm and Euglena. The only flagellated cell in human body is a sperm, which has one propulsive flagellum that is commonly known as a tail. If a man's sperm flagella are paralyzed or abnormal, the man is sterile because immobile sperm cannot reach and fertilize the oocyte. What is the structure of cilia and flagella? Each cilium or flagellum is anchored in the cell by a basal body which has 9 sets of 3 attached microtubules in cylindrical array. But in case of cilium and flagellum, that has 9 sets of 2 attached microtubules in a cylindrical array. So, each cilium or flagellum that has 9 sets of 2 attached microtubules in a cylindrical array, but in case of basal body that has 9 sets of 3 attached microtubules in a cylindrical array. Both basal bodies and centrioles, they replicate themselves. Electron microscopic study reveals that each cilium or flagellum that is covered by a plasma membrane, and this plasma membrane is continued with the cell, and their core is called axoneme, which possesses a number of microtubules running parallel to the long axis. Now, what is axoneme? Axoneme is the main extracellular part of cilia and flagella in eukaryotes. It consists of microtubules, cytoskeleton, which normally comprises of 9 doublets. So, the bundle of microtubules that is bounded by the cell membrane. The axoneme usually has 9 doublets of radially arranged peripheral microtubules and a pair of centrally located microtubules. So, axoneme that has 9 peripheral microtubules. So, suppose this is 1, 2, 3, 4, 5, 6, 7, 8, 9. So, there are 9 doublet microtubules which are arranged towards the periphery forming a ring, and 2 centrally located microtubules, and these centrally located microtubules, they are bounded by a sheath that is called inner sheath. So, such an arrangement of axonemal microtubule is called 9+2 arrangement. Central microtubules, they are connected by bridges and they are bounded by a sheath which is known as here inner sheath. Now, central microtubules are connected to peripheral microtubules through radial spokes. So, there are 9 radial spokes which connect the central microtubules with peripheral microtubules. Peripheral microtubules are also connected by linkers. Both cilium and flagellum emerge from centriole-like structure which is called basal body. So, basal body is present beneath the plasma membrane from which a cilium or flagellum originates. When examined carefully, each flagellum proves to be an outgrowth projection of cell's interior, so that it contains cytoplasm bounded by the plasma membrane. And the microtubules of flagellum or cilium, they are derived from the basal body which are present beneath the plasma membrane. As we can see in paramecium, Cilia are short cellular projections that are often organized in rows, and they are more numerous than flagella on the cell surface. So, 9+2 structure of flagella and cilia that appears to be a fundamental component of eukaryotic cells. Distribution of cilia. Motile cilia, they are found in lungs, ears, sinuses, ventricles of the brain, and organs of reproduction. What are the functions of cilia and flagella? Cilia play important role in the movement of substances along the outer surface of the cell. So, the inner lining cells of fallopian tube are ciliated, and these cells, they help to move the ovum towards the uterus. Cilia are found on many cells of the body, including the epithelial cells that line the respiratory passage. These cilia, they move rhythmically and they beat constantly, moving the waste materials such as dust, mucus, and bacteria away from the lungs toward the mouth. If the cilia, they are damaged due to smoking or metabolic problem, the irritants, they stay in lungs that may result in respiratory infections. Now, the structure of eukaryotic cilia and flagella is same, but eukaryotic flagella, they are different from bacterial flagella. Now, what is the difference between prokaryotic flagellum and eukaryotic flagellum? Prokaryotic flagellum is composed of a single globular protein that is called flagellin. And this protein wraps around in a helical fashion to form a rigid hollow cylinder. But in eukaryotes, the flagellum is composed of microtubules, and that has 9+2 arrangement. There is difference in motion of prokaryotic flagellum and eukaryotic flagellum. Eukaryotic flagellum has dynein protein that basically use ATP energy source to slide along our microtubules, creating a bending like motion. But in case of prokaryotic flagellum, that has rotating motion. Now, the source of energy in prokaryotic flagellum is proton pump, and in case of eukaryotic flagellum, source of energy is ATP pump. Now, the structure of eukaryotic flagellum is more complicated as compared to prokaryotic flagellum. Eukaryotic flagellum has 9+2 arrangement. There are 9 peripheral microtubules and 2 centrally located microtubules. But prokaryotic flagellum, that is made up of a single protein that is called flagellin, and this flagellin wraps around in a helical fashion, creating a rigid hollow cylinder. Now, what's about the actual protein which is involved in the formation of eukaryotic flagellum and prokaryotic flagellum? Prokaryotic flagellum is composed of a single globular protein that is called flagellin. But in case of eukaryotic flagellum, that is composed of microtubules, cytoskeleton. Microtubules are synthesized by centrioles found in the centrosome of our eukaryotic cells. And flagella and cilia also beat differently. Cilia typically move like arms of a swimmer. They have alternating a power stroke and recovery stroke. Both are in opposite direction, but they exert force which is parallel to the cell surface. So, this is the cell surface, and cilia, they exert force which is parallel to the cell surface. But in case of flagellum, that moves like a whip, exerting a force which is perpendicular to the cell surface. So, cilium exerts a force which is parallel to the cell surface, and flagellum exerts a force which is perpendicular to the cell surface. Investigators, they have shown that cilia also serves as cell's antenna, and they play important role in cell signaling. Now, what is the difference between motile cilia and his cousin? What are primary cilia? Motile cilia, which are involved in locomotion, they have non-motile cousin that is known as primary cilia. Primary cilia, they are present in our body. A single cilium on the cell surface that serves as cellular antenna. The difference in structure of motile cilium and primary cilium is, in motile cilium that has 9+2 arrangement, mean there are 9 peripheral microtubules and 2 central microtubules. But in case of primary cilium, there are 9 peripheral microtubules, but central microtubules are absent. So motile cilia, they have 9+2 arrangement, and primary cilia, they have 9+0 arrangement. Primary cilium has receptors on its surface that bind with specific molecules outside the cell. They play important role in many signaling pathways that regulate the growth and specialization of cells during embryonic development. They also maintain the healthy tissues. Malfunctioning of primary cilia that results in diseases, high blood pressure, birth defects, cleft palate, heart disease, cancer, and kidney failure. Formation of cilia. Centrioles, they multiply and line up beneath the plasma membrane. Microtubules, they sprout from each centriole forming ciliary projections. So cilia or flagella, they originate from centrioles. Let's watch this simulation. It shows several E. coli bacteria swimming past each other. This simulation also shows the flagella movement. To sum up, cilia and flagella. So cytoskeleton like microtubules make up two types of cellular appendages important for motion. They are called cilia and flagella. Structure of cilia and flagella. Each cilium or flagellum that has 9+2 arrangement, which is a fundamental component of eukaryotic cells. Functions of cilia and flagella. Cilia lining the fallopian tubes and they move egg towards the uterus. Cilia lining the respiratory tract that move the waste materials away from the lungs toward the mouth. And flagellated cells in humans are sperm cells. Difference between prokaryotic and eukaryotic flagellum. Prokaryotic flagellum is composed of a single globular protein that is called flagellin, and eukaryotic flagella is composed of microtubules cytoskeleton. Now motile cilia and his cousin, non-motile cilia, they are known as primary cilia, and primary cilia, they help to maintain the healthy tissue. Process of formation of cilia. Centrioles, they multiply and line up beneath the plasma membrane, and microtubules, they sprout from each centriole forming the ciliary projections. Let's watch this slideshow to review the structure of cilia and flagella.